Hi guys, this is Steve. My video is based on the research project that I did with my partner Lei. Our experiment focuses on testing the possibility of limiting fungus growth during the post-harvest storage process by using LED lights. LED lights are used because of their relatively lower weight, low cost of electricity, and that they're easy to install in chambers during transportation. The three types of fungi tested are spot blotch, 10-spot, and alternaria. A practical use of this idea can be like on horticulture commodities that are of lower quantity but are also highly vulnerable in fungal infection.
The initial step is to provide the growing environment for our fungi. The media that we used was potato dextrose agar. Then the next step was to mix it with water. Around 36 grams of potato dextrose agar was mixed with water at a combined volume of up to 1 liter, and we did this for 4 times. Then the mixture was placed in 5 different bottles. And then the bottles went through pasteurization for about 1 hour to prevent any possible contaminations inside. After that, the mixture was quickly transferred to plates when the bottles become touchable, due to the fact that it solidifies really quickly as temperature drops. Finally, all 3 types of fungi were transferred to the plates in a tissue culture lab. 70 plates for each fungus. And there they are, our lights. That's when we're installing. And here is the final setup. The little door up there had to be kept open because the air ventilation system was not very effective. So here is a general review of our experiment setup. 6 different types of light. On the top left side is one white, which means white mixed with orange. And one next to it, pure white, it means no other spectrums added. These 2 lights provide similar conditions as they do in regular labs. Beneath them are yellow and green, which are sort of assisting the 3 fungi's growth. At the bottom level are blue and red, where we had significant observations of how light can limit fungus growth. After the initial week of being exposed to light all day, the 3 types of fungi were then left in dark conditions for further observations, like this. We did this because under blue and red light, a very high percentage of fungi growth ended as early as the 5th day, and no growth the next day. So we wanted to test if the fungi were just weakened or our blue and red lights were actually capable of killing them. My part of the experiment is to focus on the light cutter's relative effects on the 3 fungi. What I'm showing here are my parts. Those that were hung the highest. This is 12 inches above the shelf. Okay, let me pick one so you guys can have a closer look. See the labels we drew that kind of radiates outward? Every label means 1 day. And this one's growth ended on March the 10th, which was the last 2nd day of our light experiment. Yeah. This one's growth, ended on the last day, which was March the 11th. In terms of growth, active examples would be the ones under green light and yellow light. Those fungi colonies have blackened parts stretching all the way from the center to the edges. These fungi colonies were tested under the medium intensity of blue light. The difference is shown directly on the appearance of those plates. Their black centers were highly limited, so as a result, the growth couldn't reach further. Our encouraging results have shown that digging more into this area is necessary, because there is also a possibility of limiting bacterial and pest growth by using LED lights. As a matter of fact, discovering harmful spectrums can be way easier than discovering beneficial spectrums. Lowering the amount of wastes is just as important as increasing yield. And that will be all I want to illustrate today. Thank you for watching.